In this video, I want to talk about neuroplasticity. Neuroplasticity refers to how the nervous system changes in response to experience. Because the nervous system isn't set in stone, it's constantly changing. For instance, when we form new memories or when we learn new things. And we have only a very limited understanding of how this happens. But at the level of the cells of the nervous system, we know a few things that go along with neuroplasticity. So one way to define this term is that it refers to changes in synapses and or other parts of neurons that affect how information is processed and transmitted in the nervous system. Neuroplasticity goes in both directions. The strength of information flowing through a particular part of the nervous system can increase, which we call potentiation. Potentiation. Or the strength of information flowing through parts of the nervous system can decrease, which we call depression. Depression. And the use of the word depression in this context shouldn't be confused with the emotional state of depression or the psychiatric disorder of depression. Here it refers to kind of depressing the responses of cells to other cells in the nervous system versus potentiating the responses of cells. The amount of neuroplasticity is highest during development of the nervous system and lower afterward, but it's still present throughout life, and it transiently increases following nervous system injury. Parts of neurons and chains of neurons that are used often grow stronger, meaning that each action potential will have a larger effect on the target cell, which we call potentiation. Parts of neurons and chains of neurons that are used rarely grow weaker, which we call depression. Neuroplasticity can happen at the synapse, which we can call synaptic neuroplasticity. Synaptic neuroplasticity, or neuroplasticity can occur at the level of entire cells, where the total number of synapses between a neuron and its target cell are changed. And this we could call structural neuroplasticity. Structural. So let's go through a few examples of some of the changes that we know about occurring with neuroplasticity. First, if we look at synaptic neuroplasticity, let's look at an individual synapse that's seeing a lot of activity and another synapse that's not seeing much activity. So that here in green will be the axon terminal of these different neurons, and here in light blue will be the target cell membrane, seeing a corresponding amount of activity from the axon terminal that it's synapsing with. So for this synapse that's seeing a lot of activity, let me just draw a little line for time and a bunch of little spikes representing action potentials. So we'll say that these are all action potentials, and there's just lots of action potentials coming down this axon, so that this axon terminal is frequently releasing neurotransmitter into the synaptic cleft and frequently stimulating the target cell by lots of neurotransmitter binding to the neurotransmitter receptors on the target cell membrane, on the postsynaptic membrane. Well, several changes can happen at the level of this individual synapse for synaptic neuroplasticity that are potentiation, meaning that each individual action potential will start to elicit a larger response in the target cell. One change that can occur is that for each action potential reaching the axon terminal, more neurotransmitter may be released into the synapse, so that a bigger response is going to be seen in the target cell because more neurotransmitter is released from the axon terminal with each action potential coming down the axon. Or the change may occur on the postsynaptic membrane. There may be an increase in the number of neurotransmitter receptors in the postsynaptic membrane, or changes to the types of neurotransmitter receptors, or the responses that occur through second messengers, so that for any given amount of neurotransmitter that's released from the axon terminal from one action potential, a bigger response is seen in the target cell, just because it's much more sensitive to the neurotransmitter that's being released. Either or of these changes, from the axon terminal releasing more neurotransmitter or the postsynaptic membrane becoming more responsive, we're going to see an increased response in the target cell per action potential that's reaching the axon terminal. So that would be synaptic potentiation. Now there's a lot of research going on trying to understand how these changes occur, because it seems like there's communication going both directions, from both the axon terminal to the postsynaptic membrane, as well as backwards, and all the processes for how this is happening have not been worked out yet. But now let's consider the opposite. Let's consider synaptic depression. So let's say I draw a little line here to represent time, And let's say we're having very few action potentials, just the occasional action potential. I'll just show this little spike here, and that we're just not having much activity. We're not having many action potentials reach this axon terminal. So basically the opposite responses that can happen with synaptic potentiation, with synaptic depression, we may see that the amount of neurotransmitter released from the axon terminal decreases per action potential, so that for each action potential, less neurotransmitter is released into the synaptic cleft. Therefore, there'll be less of a response in the target cell. And or we could see that the neurotransmitter receptors may decrease in number, so that maybe we had more neurotransmitter receptors to begin with, and that some of those go away. So we have a smaller number of receptors, or changes to the receptors to some less responsive kind of receptor, or changes to second messengers, so that the target cell just doesn't respond as much to any given amount of neurotransmitter. So with either of these changes, we'd see less of a response in the target cell to an action potential reaching the axon terminal. Now, in addition to these changes at the level of individual synapses with synaptic neuroplasticity, we can also see changes in the total number of synapses between a neuron and its target cell that we can call structural neuroplasticity. So for example, let's consider a couple of chains of neurons. So let me draw couple of neurons in a chain for each of these examples, the potentiation and the depression. And let's say they start out looking pretty similar. They both have about the same amount of dendritic branches, and the length of their dendrites are about the same. I'll just leave the dendrites off this one, and we'll say that we have about the same number of axon terminals coming out and forming synapses between this neuron and this other neuron, which will be its target cell in this situation. Now I'll just draw a little axon on the target neuron as well. So if these two neurons are firing together frequently, if this neuron is firing lots of action potentials and this neuron is firing lots of action potentials in response to this neuron stimulating it, we can see an increase in the number of synapses between these two. And we can see that from the dendrites. We could see the dendrites getting longer or growing more branches, so they become more complex trees of dendrites. Or we could see that from this presynaptic neuron, it could start sprouting more axon branches and terminals so that it's forming more synaptic connections with the dendritic tree over here. So with this kind of structural potentiation, both of these neurons are sprouting lots more little branches. They're either sprouting axon terminals or sprouting more dendritic branches. So I'll just write that down here, that we're doing lots of sprouting. Just like plants may sprout lots of new shoots in the spring. And then the opposite may occur here. If we're not having very many action potentials being fired by this neuron or by this neuron, and particularly if they're not firing action potentials together, we can see the opposite, where we actually start losing length of dendrites or losing dendrite branches, and the dendritic tree can become simpler and shorter. Or we may start losing axon terminals. We may simplify the axon terminals that are coming out of the axon. And if this neuron is not firing very often at all, we may actually lose this neuron. It may actually go away. And this type of structural depression, where we're actually losing parts of neurons or entire neurons because they're not very active, we call pruning. Again, just kind of like plants, if you're kind of pruning pieces off a plant so that it has less twigs or branches, it's the same idea. Now, both potentiation and depression can happen over a wide spectrum of time, and we often kind of divvy it up into short-term changes, such as on the order of seconds or minutes, or long-term changes that can be months, years, or even decades. And synaptic neuroplasticity can contribute to both short-term and long-term potentiation or depression. And then these structural changes tend to go along with more long-term potentiation or depression. And I think you could imagine how by changing the strength of information flow through individual synapses or between cells, by changing the total number of synapses that there are, that neuroplasticity can play a very important role in development of the nervous system as it's wiring itself together based on the experience that the nervous system is receiving during its formative time. And also this plays a huge role in memory and learning and recovery from injury to the nervous system when it's kind of trying to wire itself back together after it's been injured. So these are a few of the things we know about neuroplasticity, but there's a lot more that we don't understand yet. And there's still a lot of research going on trying to understand how all these processes happen and how they contribute to all these amazing functions of the nervous system that can change over time.